Today, we'll talk about the program and some of the topics that we will be covering. We'll also discuss some of our previous experience with these kinds of training programs in bioinformatics, and hopefully answer any questions that you have about the upcoming program. So OmicsLogic Transcriptomics is an online training program for students, researchers, and clinicians, and it is designed to introduce participants to the various aspects of RNA-seq data analysis in application to basic research, as well as in biotechnology and biomedical applications. So what I hope to do today is speak a little bit about how RNA-seq is used for real-world biomedical applications and research projects, and what kinds of skills you need to have to be able to handle large RNA-seq data and be able to visualize, analyze, and interpret this data to study different types of biological phenomena. A little bit about OmicsLogic and our programs. So the OmicsLogic programs are a result of a close collaboration between Pine Biotech, our company, which is based in the US, and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. As a result, the OmicsLogic programs have been thoroughly tested and are known to lead to excellent outcomes for students and faculty interested in learning bioinformatics or incorporating bioinformatics resources into existing programs. These programs combine experience in bioinformatics and terminology of biological data, as well as data science skills that are in high demand in industry. We've been testing these programs for several years now, and we feel that the outcome of these programs is very encouraging because many of the participants that we have joining these programs are continuing on to apply the skills that they've used to their own research or even in industry. So what is omics? Omics technologies like next-generation sequencing can be used to explore the roles, relationships, and actions of the various types of molecules that make up the cells of an organism. For example, this is an image of a cell, which gives us phenotypic data about what these cells look like. But many types of omics data can actually reveal molecular detail about what is going on inside the cell at the subcellular level. For example, genomic data tells us about variants at the DNA level. Epigenetic regulation of gene expression has been of an increasing interest for the past several decades, and today we can study this kind of epigenetic regulation by analyzing chromatin immunoprecipitation data, as well as by sulfide sequencing data and the expression of micro and long non-coding RNAs. But ultimately, we want to understand how the cell, which functions and grows, is regulating the different activities based on expression of mRNA, messenger RNA. That could be studied through alternative splicing, how different genes are spliced together by combining exons, as well as their level of expression, which could be studied at both tissue and single cell level. So what we'll do in this program is we'll talk about some of the basics of RNA-seq, how processing, mapping, and quantification of RNA-seq data, how to explore the gene and isoform expression data, and how to prepare the data for analysis, what type of normalization techniques and data pre-processing techniques make an impact on our ability to interpret what is going on at the subcellular level. We'll discuss some of the statistical approaches to analyze differences between conditions and identify statistically significant features of our data sets, how we can then look at what these features mean in terms of their biological activity, and how we can combine them into gene sets and pathways. And finally, we'll talk about machine learning. So we'll talk about how the advances in machine learning and the easy application of machine learning made through different packages and interfaces can allow us to study detailed patterns within this large RNA-seq data so that we can interpret differences between cell types, tissue types, and different types of conditions. As a method of measuring RNA translation, transcriptomic studies can help detect mRNA expression inside the cell as an accurate alternative to membrane protein expression detection, which allows for molecular precision in diagnostics and even development of treatment based on molecular pathways. So when we think about RNA-seq, the utility of understanding what types of proteins are being produced provides us with a clear understanding of what happens inside, beneath the membrane of an individual cell, which is very different from when we talk about staining for proteins in biomedical and diagnostic applications, and also provides us with a good insight about potential targets that could be targeted by different drugs. As an important part of precision medicine, this type of information is becoming regularly useful in different types of diagnostic purposes for early detection, specific and personalized understanding of what's happening with a patient at the subcellular level, what is driving the disease, and exploratory analyses with model organisms and cell lines where functional association between different genes and variation in those genes could be understood and studied at a mechanistic level. So to allow us to study this kind of technology, over the past few decades, several different technologies have been designed. And RNA-seq refers to this next generation sequencing technology. So RNA-seq or RNA sequencing has revolutionized the way biologists examine transcriptomes and has been successfully applied in biological research, drug discovery, and clinical development. Compared with microarray-based transcriptome profiling, RNA-seq has a wider dynamic range, meaning that it can detect many more variations of the same gene, so many more transcripts of a gene, because it is not based on hybridization. So it's not based on probes designed to measure and detect specific types of mRNAs. RNA-seq can measure the expression levels of thousands of genes simultaneously and provide insights into functional pathways and regulatory networks in biological systems. In addition, RNA-seq can provide novel insights into alternative splicing, unannotated exons, and novel transcripts that have not been previously annotated. Most importantly, such detail has already proven to yield significant discoveries in many research areas. Gene expression patterns in association with clinical data. So when we have both the gene expression data and different types of phenotypic data described by what condition this is, or maybe even described in a continuous numeric data, like for example, LD50, can help identify molecular biomarkers for diagnostics and therapy selection, like biomarkers. And these new discoveries also include a better understanding of complex diseases, for example, like cancer, that include aberrant expression and changes in isoforms or exon splicing. And this understanding of these complex diseases ultimately leads us to appreciation of the variability of the disease at an individual patient level. It is also helpful to identify targets for novel drug design and repurposing, and requires an understanding of both the data preparation, how the data is stored and annotated, and ability to find associations and measure them to be able to address these important challenges. So in this program, we'll discover how RNA-seq data can be used to study the role of tumor microenvironment in tumor growth or regulation, which could be studied for differentially expressed genes in different types of conditions when we compare different conditions or control experiments, and analyze to find markers of cell and tissue types in neurodegenerative diseases. So we've prepared curated data sets and projects that we will be able to explore as we learn about the basics of RNA-seq data. So this program, OmicsLogic Transcriptomics, includes 6 sessions that are interactive sessions with a mentor. They also include access to a variety of different resources that you will have access to during the program, and an ability to work on an independent project. So let's take a look at some of these sessions and the topics that will be covered in these sessions. In the first session, we will describe all of the tools and resources for transcriptomic data analysis. So we'll discuss a lot of different material that was prepared for the program specifically and how to use some of these tools. In the next session, we'll discuss the basics of processing next-generation sequencing data using what's called bioinformatics pipelines. So we'll discuss the concept of pipelines, and we'll also discuss specific types of pipelines that can help us generate different types of transcriptomic data at the gene or isoform level, including bulk and single-cell RNA-seq. Then we'll discuss analysis, normalization, and visualization of gene and isoform expression. We'll do this using both cloud-based tools that are intuitive and easy to use to understand the logic of analysis, and also discuss some of the typical packages in both R and Python for analysis and visualization of such data. In session 4, we'll introduce big data mining and machine learning for predictive analysis. So we'll discuss how the emergence of big data in such studies like RNA-seq has led to an ability to mine publicly available data sets, find interesting associations, and develop novel hypotheses. We'll also talk about how machine learning allows us to deploy machine learning predictive models to learn from data that is already available and predict something new about novel data sets or data sets from other repositories that could be very useful for classification, early detection, and development of biomarkers. In session 5, we'll speak about the analysis of single-cell RNA-seq, how we can analyze single-cell data, so what kinds of pipelines and standard packages exist for the analysis and the processing of that data, what are some of the challenges associated with RNA-seq data, what are some examples where analysis of single-cell data have proven to be insightful and important, and also discuss tissue-specific expression. So in fact, how we can take markers from different tissues discovered through single-cell RNA-seq and apply decomposition of bulk RNA-seq data. In session 6, we'll take a look at a few hands-on project examples, and these will be from the domain of cancer biology, infectious diseases, and biotechnology. So we'll provide a few different examples, and using these examples, you'll be able to develop a project of your own. And in that project of your own, we will guide you how to find the data, how to then process that data and prepare it for analysis, analyze and prove or disprove a hypothesis, and then visualize and showcase the results of your analysis in an impactful way. Now you can see on the chat, there's a link to a page where you can find all of this information. So all of the questions about what topics and sessions are included, as well as the course costs and different options are described on this page. So let's take a look at this page together. So this is the page. Thank you, Gupta. Okay, so all you need to do is go to transcriptomics.omicslogic.com. So this is the page. You can see that it's going to start on 15th of September. We have 4 days and 8 hours, and we're counting down to the beginning of this program. And you can also start looking at different sessions and topics that are going to be covered in the program. and also the calendar of those sessions. So essentially, we're talking about 2 weeks. In these 2 weeks, we will cover a lot of the basics in the first week, and then go on to more advanced topics in the second week. And then you will have another 2 weeks to develop and work on your independent project. Now, in many cases, when we interact with participants of our program, they are interested in having a mentor guide their selection of a data set, help them develop the project idea, and even help and assist to guide their analysis. Now, we do have that option available as well. So as we thought about how to structure this program, we were thinking about different packages that we could provide to participants that could benefit the most from completing a program, getting certification, developing a project idea, and actually applying some of these skills to a data set or a research question of their own. So that's why if you go up here and you click on Get Started, you will see a few different options, which includes access to the program courses that we will take a look at in a second, access to our coding exercise platform where you can code, learn how to apply different packages, and also get help and feedback on some of your code or errors that you're getting, access to the interactive sessions where we discuss in detail some of these topics and answer any kind of questions or discuss any technical challenges that you have. Also access to research projects. In other words, you'll be able to analyze your own data and get guidance on the analysis of your own data from a technical perspective, and optional mentor support. So that mentor support also means that you'll be able to interact in a one-on-one session with a mentor, ask any questions, get your answers, and also be able to use that help to make your project better refined and more ready for prime time for others to see. So those are some of the choices. Now, if you scroll down, you will see, as we mentioned, some of these topics explained, and here you'll see some of the benefits from these programs. Also down at the bottom, there's some feedback that we've gotten in the course of our previous programs that you can take a look at, and the description of the coursework that we will go through in detail in this presentation. So the OmicsLogic programs include a variety of resources that you can benefit from and also be flexible with your schedule, making the most of your time. For this program on transcriptomics, we will combine online sessions, self-guided study materials that you can complete on your own time, and practical assignments. And as a result, we hope that everyone will be benefiting from an immersive experience. And what we've seen in the past is that the most of these elements actually work seamlessly together to have an effective outcome for our previous and ongoing OmicsLogic programs. And we have a few others that are ongoing as well that I'll be able to share in a little bit. So let's look at some of these resources one by one. Throughout the program, you will be accessing some of these online asynchronous resources that provide a broad and yet detailed overview of processing, statistical analysis, and machine learning tools, as well as understanding of single-cell data types and analysis techniques. The courses cover methods, expand on the specific terminologies associated with each type of analysis or data, and provide practical examples. They also include quizzes for self-evaluation and assignments. So let's take a look at some of these courses and what they include. Okay, so this is a link specifically to the different collections of courses. Throughout the program, you'll have access to many of these courses, not all, but many of them. But specifically, we will focus on this collection of transcriptomics courses. So you can read here about each individual course and what it includes. As a part of the program, all of these resources are going to be free for you. And they actually fit in very nicely into the program schedule that we have looked at already. So in Transcriptomics 1, we discuss what is next generation sequencing and how is it different from first generation sequencing. We also talk about the biology of mRNA expression and how it could be related to different pathways and signaling and metabolic pathways at the subcellular level that we need to or seek to understand by analyzing such data. In Transcriptomics 2, we turn to statistical analysis. So we discuss how statistics has been utilized to understand the differences between conditions or groups of samples, and what are some of the considerations around doing that kind of analysis and interpreting the results. We also talk about annotation, biological annotation of pathways and gene ontology terms, and think about different applications in a variety of directions. In Transcriptomics 3, we turn to machine learning. And machine learning, as you know, is divided up broadly into two types: unsupervised or data mining, and supervised or classification. So we discuss a few examples based on some practical applications in the context of precision medicine. How can we understand and classify, build a classifier that can predict different types, subtypes of cancer? In Transcriptomics 4, we turn to single cell data. This is a new technology. There are still different methods in development, but there have been some established pipelines that are now being standardly used to process and produce a table of single cell gene and isoform expression data. Because that data comes from a very different type of data preparation method, you also have to have a number of considerations when you look and analyze the patterns that have been made through that kind of a processing pipeline. So that's what this course is all about. In addition to the online courses, this program is designed to give you a sense of practical analysis tools. And we want to make this as approachable and as useful for a wide variety of participants, both those that do have some technical background like coding and those that do not. But most importantly, we're going to be analyzing big data sets. So these are going to be tens of samples at a time. As you know, a single sample of RNA-seq data could range anywhere between hundreds of megabytes to a number of gigabytes. And so processing such data sets on your own computer is going to be time-consuming and ineffective. So as a part of the OmicsLogic Transcriptomics program, you will have access to the Bioinfo platform that provides an interactive learning experience. The platform is color-coded to help establish a logical connection between the theoretical aspects of data analysis methods and the practical steps to achieve a biologically interpretable results. So this is what's called the bioinformatics pipeline. In this pipeline, every step of the way, you will get an informative pop-up that explains the logic of why we are performing a certain step, discusses the inputs and outputs of every step along the way, and guides your processing so that you can understand what are the typical mistakes that you can avoid by focusing on the correct method for the correct data set with the right type of an output in mind. As a result, instead of spending time on building the pipeline, you get the ability to practice the pipeline application, different methods with different types of settings, produce different types of outcomes and results. And so you will benefit from making those results throughout the program and being able to explore how these results could be interpreted from a biological perspective. Now, what's important is that many different pipelines are designed to produce different types of outputs. And we can go to a very detailed read level analysis where we can understand the mapping quality and the read alignment for different types of data that is being prepared, to then understanding the outputs or the gene expression table, gene or isoform expression table. We'll talk about how to read these different types of outputs, visualize them, and also perform analysis to summarize and explore the specific patterns within the data that we prepare. In Transcriptomics 3, we'll spend a significant time to understand machine learning or data mining and classification. With the exponential growth of data, many are turning to machine learning to study complex relationships at the subcellular level and develop data-driven research with important outcomes for human health. So in this program, we'll have a chance to see machine learning in action, including data mining techniques like clustering, dimensionality reduction like principal component analysis, classification like random forest and support vector machines, and feature selection algorithms that are both stepwise, greedy, and selection-based. So we'll cover a wide variety of topics that are related to both processing these big data sets, then analysis from a statistical perspective, and understanding how these more complex machine learning methods are practically applied in really clear examples, which I'll talk about in a second. So let's take a look at how this process will actually work. During this program, you'll have access to the Bioinfo platform. Now, all of you, whether you're going to be a participant in the program or not, are welcome to try this platform by requesting a demo account. If you do request a demo account, you'll immediately get access to the interface where you can explore a variety of different sections, including next generation sequencing data, mass spectroscopy, structural biology, and data integration and modeling. Let's take a look at the RNA-seq section to understand what it will look like. Here we've prepared a number of different examples. You can see from cancer cell line projects, Angelman disease, which is a neurodegenerative disorder, and patient-derived xenograft models to understand the effect of microenvironment changes on tumor growth. Inside the interface, you can now select different methods to build a pipeline. It all starts with the start. And here you will see how different types of methods that are described in the further sections are used for what types of applications. You can then see that only a small number of options are highlighted. And so based on your preference, you can select pre-processing steps that clean up the reads and help you focus on only high quality reads, eliminating contamination from such things as adapter ligation and PCR amplification. Then we can look at mapping techniques to the reference genome or transcriptome and develop a sense of how high is the expression for a particular gene or isoform, ultimately looking at the gene expression table. So once this pipeline is complete, you'll be able to see some of the output files, and we'll discuss downstream analysis. I'm going to pause here. I see that there's a few questions, so I'll just briefly address those questions. If you have any questions about what I've shown so far, please put them in the chat. Okay, so how will all of these topics be covered in 6 sessions only? As a result of combining the interactive sessions, which are about 1.5 hours long, together with the asynchronous material that you will have access to on your own, we expect that you will be able to get a very good understanding of many of the topics that I'm discussing here, and also try to apply them to an independent project. However, the whole program is about a month long. So if you don't have a lot of time on your hands, you will have additional time after the interactive sessions to go through the synchronous material and review the recorded sessions, which will be posted on the forum, so that you can make the most out of the time after the interactive sessions have been completed. Okay, what is the RNA-seq pipeline? So what you're looking at now is the RNA-seq pipeline. A pipeline means that data from a script or a job has been submitted to the next script, and in a sequential manner, the pipeline produces outputs that are used by the next consecutive method to give you the final output. So this is called a pipeline. Now, beyond this pipeline, which seeks to produce structured data from unstructured data, which comes out directly from the sequencer, we will also look at how to analyze such data. For example, we can look at the data in the gene expression table to study different patterns and even visualize them. Because there's so many elements to analyze, this could be done in an automated way using different types of scripts. Okay, about my voice breaking, I'm really sorry about maybe the connection quality is not that great. And I think if anyone else has this issue, please let us know. We'll also provide a recorded version of this webinar.
We can hear you.
Is there any chance
Video.
you can hear me fine?
Yeah, we can hear you clearly. Thank you.
Okay, great. Is there any chance of publishing the paper or data in case of research project package? Yes, the objective for those that select the research package is to provide you with an opportunity to develop an independent project and make sure that that project gets to a level of completion when you can actually analyze the data and showcase what you have discovered. Now, as you know, publication itself requires additional steps like peer review and replying to different questions from your peer review process. So we're not going to guarantee that you're going to be able to publish, but we have definitely seen a number of participants that have been able to achieve meaningful results. And we even have a few examples on our on a page. Maybe Vipsa, you could paste a link to the project description with a few examples of participants that have made excellent projects. Sometimes these projects take a longer time. And so we also have an internship, or what we call a research fellowship program, that's intended for 3 to 6 months. And during that time, you have plenty of opportunities to interact with mentors and a community of like-minded bioinformaticians that are going to help you make the most out of your independent project. So Bipsa, if you have the link to the internship page, that would be also useful right now. So another part of what we will do is use a coding platform. Now, coding itself in both R and Python is very meaningful and useful for anyone that is interested in bioinformatics. Now, some of you might already have some skills in basic R and Python, but what we seek to provide in this program is an opportunity to learn, apply, and combine together different scripts to make the most out of the data. So let me show you how that would work. Throughout this course in the program itself, you'll have access to tutorials that will explain how to load data and check the different data types, perform exploratory analysis, doing descriptive statistics and visualization, and then focus on dimensionality reduction, differential gene expression, hierarchical clustering, and different methods of classification. So as you go onto this platform, you'll find that inside, you will have some explanation in both R or Python on how to perform these steps, learn about different types of code and syntax, and then apply this to a data set that is described in the program itself. So when you learn how to do this, you will actually be able to run a code, you will get errors or success, and also will be able to monitor and record your progress as you go through this process. So for example, here, I have just prepared some summary plots that allow me to understand what my data looks like. And by changing some of these examples, I can also modify how this is going to look in the end. So for example, here you can see I can change the colors or change which data set I'm going to be using And for example, let's just change briefly this code from red to blue. Okay, and you can see that we got a blue box plot. So those are the types of exercises that you'll be able to go through. And this is also going to be included for a whole month. And after that month, you'll have a discounted subscription option if you're interested to continue and learn not only these things in the context of RNA-seq, but also our full data science curriculum. Most importantly, I believe that you actually understand the most of the significance of RNA-seq and understanding gene expression from a biological perspective when you get to apply this to data sets that make sense, to data sets that you understand the challenge and where you understand what the data was prepared to do. So we'll take a look at a number of interesting examples taken from high impact journals that are peer reviewed. We'll take the data and we'll also discuss how to find such data on repositories like NCBI and look at them in the context of different types of applications. For example, for cancer, we'll talk about patient-derived cell lines, patient-derived xenograft models, and clinical trials and translational applications in repositories like the Cancer Genome Atlas, or TCGA. In depth, we'll go into some examples where we discuss what are the challenges. For example, in breast cancer classification, it's important to rely on molecular subtypes to discuss different treatment options and risk associated with different types of therapies. So we'll talk about how these different subcellular profiles can help us identify molecular classes and what do those classes mean in the context of diagnostics and treatment. Can we download the sessions and other inputs for a future reference? Of course, we will provide you, and I will get to that in a second. We will provide you with recorded materials from all of the sessions. You will get access to all of our course material that is also available in a PDF format. And you'll also get access to the data sets that we have practiced and used throughout the session. So you will have that after this program is over. One of the projects that we'll discuss is modeling precision medicine in cancer. So here we will take a paper that is called Modeling Precision Treatment of Breast Cancer, where a number of different cell lines were prepared and accessed by the researchers from the public domain, and developed a classifier that accurately predicts how responses to different treatments will be based on the molecular profile, but also by using training data from response and measurement of inhibition of growth 50, so 50% of inhibition of growth. As a result, if you look just through the article itself, you'll see how we will learn about molecular profile, what are these features, how to predict therapeutic responses, what are optimal predictors, what is molecular pathobiology, omics data, and how to select most predictive molecular feature sets. So you'll get access to a number of projects that are really broken down and clarified in terms of what is the objective, what is the data, and how to apply the different methods of analysis to discover such important patterns and be able to speak about them and present them to your peers. Now, many of our previous participants have been able to leverage these programs to then go on and start discussing the projects that they have worked on in the context of their future journey. So here you can see some of the previous participants of our programs that had a chance to present at conferences. And by the way, I want to mention that right now, for these kinds of online programs, this is a good time because you can actually present virtually in many conferences that are upcoming. If you are interested to publish, a poster session is probably going to be your first step to validate and get some feedback on your idea. After that, as you develop your passion in a particular area, or maybe you already have, maybe you're already using RNA-seq and data analysis skills for a particular type of projects, you can then build on your expertise in other areas to advance your research career. For example, we have seen a number of participants that have come from either bachelor degree, master degree, postdoc, or PhD, or postdocs that have been able to benefit from the program. For example, Olga Udartseva from Roswell Park Comprehensive Cancer Center mentioned that the program from her perspective is very well structured and easy to learn. The skills that she gained through the program helped me significantly enhance the relevance of my project by making connections between my preclinical study and its practical applications to cancer patients. So we're not just talking about learning about RNA-seq. Our goal is to empower you to then apply the different bioinformatic tools, repositories, and data sets to actually make research discoveries and apply them to practical problems. Now, she's not the only participant that have provided with such feedback. We believe that many of the participants that we have had in our previous programs have been able to benefit from these programs. And as a result, we have them available both in this online fashion where you can just join online, or through different universities. For example, in Louisiana, we are offering such training through the Louisiana Biomedical Research Network. In India, we have partner institutions that are able to provide these courses to their participants, as well as a new program that we are launching in Africa with a number of partner universities that are going to offer such coursework and resources to their students. So the program is going to be starting on September 15th. So that's just in 4 days from now. Remember that you'll have access to processing pipelines. We'll discuss different methods of visualization and how to build that visualization using different types of packages, coding skills, as well as tools. We'll discuss statistical analysis approaches, how to do hypothesis testing, and how to interpret the results. We'll discuss data mining. How do we mine the complex big data sets that are available online with thousands of samples, and how to extract meaningful patterns and meaningful associations from that kind of data. And then we'll also talk about interpretation. What are some tools that can aid us in visualizing our results and being able to interpret them by using different reference databases for annotation. At this point, I would like to ask if you have any questions or if I could clarify any part of the program itself. The OmicsLogic Transcriptomics program will allow you to learn about RNA-seq data processing, analysis, and interpretation, leverage cloud-based tools for hands-on assignments, apply machine learning to large biomedical datasets, complete and present an independent research project, and obtain a certificate of excellence and gain recognition from our community. And if you have any questions, please paste them on chat. I want to briefly mention how the program is structured. And for those of you that have already registered, I know that many people have already registered for the program. So just to explain what would be the next steps. Once you are registered, you will gain access to our program page. And this is the program page, OmicsLogic Transcriptomics 2020. Here you will see courses that are included in the program, project examples, progress where we track the progress, and you can also evaluate your progress from a perspective of the rest of the group, track assignments, and also see the syllabus and the faculty of the program. We also utilize the forum. On this forum, we will be pasting session recordings, highlighting top performers, and also pasting any additional information that we think is beneficial to those that are interested in RNA-seq. So these are going to be publications, different types of review articles, and data sets that we are utilizing in the program. About financial support, we are very interested in helping anyone who is passionate about learning bioinformatics gain skills and apply them in a meaningful way. We offer scholarships for those that have the time and the dedication to go through these programs and develop an independent project. You are welcome to reach out to marketing@pine.bio with any clarification about offers for a scholarship. So you do have to have some previous background in either molecular biology or some kind of bioinformatics so that you are comfortable with the materials, and you have to be, please email marketing@pine.bio. So on this page, you will see all of the relevant materials. This page is going to be available to those that have already registered. You'll see the upcoming schedule for each one of our sessions. And you'll also see any kind of activity here and get help from our mentors to address any technical challenges throughout the program. After completing this program, can participant apply for scholarship programs? You can definitely apply. There's a variety of different scholarship options. I also want to mention that we do have a LinkedIn group and a Facebook group that we invite you to join. Maybe Deepsa, you could post those links. In those groups, we regularly post opportunities for different types of internships or even job offers. In terms of scholarships, so as I mentioned before, we do have a program for internships. Let me just open that one, which is a research fellowship that is either 3 or 6 months, conducted completely online for those of you that might be interested in that. In the research fellowship, you gain a lot of assistance from like-minded bioinformaticians as well as from our team to help you develop an independent project and complete it in the course of time that you're registered for. And we believe that if you're interested in a scholarship program or you're interested to get into a particular program or a lab, typically what is required is for you to come prepared, right? A lot of PIs, a lot of universities are looking for students that are prepared, that are ready to start their job or start their position running. And so we believe that developing a project in the specific topic of that lab or that university or that company can really help you advance your career in that field. So we do offer support and access to a lot of the tools and regular weekly sessions that you can join here. You can, again, fill out the form on this page if you're interested specifically in this. You get access to all of our learning materials, and by filling out this registration form, you will start receiving emails about the specifics and the details, and we can also schedule a one-on-one meeting if you're interested in joining that program to evaluate the best fit and discuss what you might want to do to prepare for such a program. Okay, so if there are no other questions, I would like to also introduce Dr. Mohit Mazumdar, who's going to be also helping with this program. Mohit, if you would like to say a few words about the program or anything you think might be relevant at this point.
Thank you, Ilya. Hello, everyone. Yes, for this transcriptomics program, which will be starting on Monday, we will be helping you out with the registration on the website, on the portal, on the educational platform, and give you the access to the courses that we described today in today's session. And we will have we'll be also sending you the access to the T-Bioinfo server account so that you can start applying those methods and see how you can design those pipelines as well. So if you need any help regarding the registration, do let us know. If you face any trouble, reach out to us at marketing@pine.bio. And we'll be looking forward to have you on Monday. Yeah, thank you, Ilya.
Okay, excellent. Thank you, Mohit. And thank you everyone for joining today. Some of the questions, do you get a discount if you registered today? So please reach out to marketing at pine.bio. You will see when you get started, there is an option here to get a coupon. So if you are interested in a discount or a scholarship, you need to discuss that with marketing at pine.bio. We review every case with a lot of consideration. We're really interested in more than just a program that you walk away with a certificate from. We're interested in building a community that continues to learn together and actually applies these skills to different types of research projects. So we're definitely happy to have everyone who's passionate about bioinformatics on board. Thank you for joining today. Again, the recording of this session will be made available. There are other programs, but all of them are in bioinformatics. We do discuss aspects of cellular biology, immunology, infectious diseases, etc., but in the context of data analysis. And we are very, very passionate about the data part of each one of those topics that you've mentioned. And in fact, what we try to do in our sessions is not only discuss about the data analysis methods and the statistics and the computer side of things, but we also touch on many aspects of cellular biology, immunology, and other topics that are important to understand when you deal with biological projects and biological data. Any courses in cancer biology? So in the courses that we have, we include introductions to different types of projects. So when we look at the, let's look at the schedule here. We believe that in the beginning, you should get some basics and be able to independently analyze data. And so that's what the first part of this program is going to be focused on. Then we'll actually discuss a number of project examples and how to find publicly available RNA-seq data. So a lot of our examples are going to be related to cancer, both cancer biology and immunology, in the sense that the cancer develops and then causes disease in different areas of the body. We do have a separate program on oncology, which is not this program. This is specifically on transcriptomic, but transcriptomic data is excellent to study cancer, particularly different types of cancer like breast cancer, etc. But we also apply the same to the immunology of infectious diseases when we look at the host response to different pathogens. And we also discuss different types of approaches to agriculture, understanding of differences in crop production, stress responses, and others. So if you need a little bit more information on the different types of projects that we offer or that we discuss in the context of the program, you can take a look right here. These are some of the projects that you will have as your practice throughout the program. You can see here that we have identification of novel pathogens in cell lines that are used for biologics production, Ebola virus. Some of these are not going to be available for the whole public, but we will still rely on them in the courses. Crop drought stress and resistance, targeted medicine or cancer biomarkers in early stage detection of breast cancer, neurodegenerative diseases like Alzheimer's disease, analysis of changing immune responses in cancer. This is a study of macrophage changes over time. Patient-derived xenograft models allows us to study the microenvironment and tumor development. And modeling precision medicine, which I already touched on in this course. Now, we do have a number of other examples that we're going to be doing, including liver cancer, tuberculosis, as well as COVID-19 immunology. So there's a lot of material that is prepared. Obviously, in such a short time, we're not going to be able to cover everything, but we will provide you access with some curated data sets and explain some of the basics that will get you started. and help you develop an independent project idea. And whether you select the package with the mentorship or not, we'll try to work with you hands-on to actually allow you to get the most out of the program. Okay, so again, thank you everyone for joining. It was a pleasure to see so many people interested. About the pre-requirements, there's not really any major pre-requirements to just join the program. You do have to have some basic, well, so to join the program itself, you do need to have some basic terminology in molecular biology. In fact, what we recommend for everyone, we have a free course that is called Introduction to Bioinformatics. Oh, sorry. Introduction to Bioinformatics that we recommend for everyone to start with. This is a free course, so you can actually join and register today and go to this Introduction to Bioinformatics course. Now, this course is not your standard Introduction to Bioinformatics where you learn the very basics. It talks about different types of applications, different types of projects, different types of data sets. It talks about many different things in a very short period of time. And you can see, just based on the reviews, how many different people have been able to benefit from completing this course. Now, after you go through this course, you will also get a sense of what these courses in general are going to be like. And you'll see that even faculty members at different universities were able to benefit from just completing this free course. So it's a free resource to get started. We also get to learn about you in this course, and that helps us adjust our programs to make sure that they are providing great outputs for those participants of our programs. So you can really try to explore the materials here, provide us with some information about your background, your educational background, your other experience in bioinformatics, and we will be reaching out to you based on your feedback from the course material itself, but also based on your information. Okay, so hopefully I've been able to answer all of the questions that you might have. If you do have any other questions, feel free to reach out to us by email or complete this Introduction to Bioinformatics course for free and provide us with some more information about you. We would be happy to stay in touch as well. Okay, and we also offer a number of other programs that are coming up. Briefly, those are programs in different parts of the world that focus on the application of bioinformatics to different types of data, as well as to different types of challenges, biomedical, epidemiology, as well as agriculture. So please stay in touch. We do about 2 programs per month. And if you are interested in a particular topic, we're always looking for new ideas and to develop new programs. Thank you and have a good rest of your day.